So I will talk in this first is some introduction of the microplastics. So let's see the history of plastics. So plastics was first invented in 1862 by Parkes in UK. At that time is used as celluloid, and 7 years later is the Hyatt in USA also finished to synthesize the celluloid, and then the first fabric for the production of polymers. And then from the beginning to now, this plastics underwent 3 different stages. The first one is natural polymer processing stage, like Parkes and Hyatt, just is a modification of the natural polymers. And then from 1907 to the 1950s, there is the second phase of the plastics is the synthetic resin as an industry. And during this phase, many polymers are invented. And after this 1950s, a lot of the plastic have been used in a lot of areas, like in automotive, in furniture, in IT and telephone, and also in nanotechnology. And since the 1990s, the bioplastics was in use. So this is a fast growing period. Until now, totally almost 19 billion of plastics have been produced in the world. And from the 1950s to 2018, and this production of plastic increased annually 128 million tons. And among these produced plastics, only 10% was recycled, and the 20% was incinerated, and the rest 78% was released into the environment. So there are many kinds of plastics. Here in the left is the different kind of plastics typically used. They are CC binding, CC backbone plastic is not hydrolyzable, and there are also some hydrolyzable plastics. So this is their use in a different area, and we see here most of the plastics was used for the packaging and also for the building and constructions. So here is the structure for polystyrene, PS, that I will introduce in my presentation later. So at the beginning, the people thought with the plastics, they are very happy. They can live in such kind of 3RV living. And here is a photo in the last century, 1950s. And since then, a lot of plastics was thrown into the environment, into environment. As I have said, is 79% into the environment. And this debris of plastics pollute the environment, and most of the plastics are single-use plastics. So when they're into the environment, then they underwent a lot of processes, and these processes are aging processes. They include physical, chemical, biological deterioration. What is it? Okay. Yes, laser point. I have 2 pointers here. Okay. Yeah. So here, the plastic debris can be disintegrated into microplastics, as MPs, and these microplastics. Okay. This microplastics can be disintegrated into nanoplastics, and then further degraded to DON, dissolved organic matter, and then completely degraded to CO2. So this aging process can include external mechanical force and abrasion. For example, this is a microplastic found in the sound, and also here the animals, this Antarctic clear can break down the microplastics. And in the environment, we have also on the UV radiation. This UV radiation can cause the photo-oxidation of these plastics. We have ozone. Ozone can also degrade plastics. And also, they are all chemical degradation. And also, physically, if these plastic in the water and the water is frozen, then this cryo-degradation could happen. And also, plastic can be degraded. And also, in the high temperature, with the water, this hydrothermal degradation could happen. And this is also a kind of aging process. And also some animals, especially the mealworms, can degrade the plastics. Like here is the mealworm growing on the steel pool, is polystyrene. And also some bacteria, for example, bacillus and also micrococcus are isolated and they can grow on plastics. But these plastics, small plastics, the nano-sized plastics, MPs, and the micro-sized plastics, MPs, and the nano-sized plastics, NPs, are also secondary plastics. So that means they are produced in the environment. So later I will use MNPs for NPs plus NPs. Except for the secondary sources for the MNPs, there are also some primary sources for the MNPs in the environment. So the primary MNPs are manufactured for special applications, and then they are released into the environment during the application or during production. For example, in personal care products, we have the MNPs. They are used for cleaning in the sunscreen and cosmetics. Also in the paints for the buildings, there are many MNPs, and the microfibers from synthetic textiles. These microfibers will be released if they are washing in the washing machine, for example. And also many plastics can be released from these tea bags. This is the first report in Canada by professor in Canada. They reported that these silk tea bags can release many small plastics, and also in nano size. And also electronics is a source of primary MNPs. And in the biomedicine and for the drug delivery, they use also MNPs. And the last one, but not least, is the rubbers of vehicle tires. This is the tire wearing particles. They are also in small particles. They are also MNPs. Of course, also the footwears can also produce MNPs. So there are some discussion about the definition of microplastic MPs and then nanoplastic MPs for the size. How is the definition? So for the microplastic MPs, normally we think that if this size smaller than 5 millimeters, then it's microplastics. But in some literature, the people said it's smaller than 1 millimeter. And for the nanoplastics, some people said it's smaller than 1 micrometer. But some scientists said this nanoplastic should be the same as other nanomaterials. That means the size of this nanoplastic should be also smaller than 100 nanometer. But now in the scientific, most of the scientists think that for the nanoplastics, the upper limit for the particle is 1 micrometer. It's different from these nanomaterials. So in the environment, the nanoplastic and also other nanoparticles is not easy to be detected. But some people have detected these nanoplastics in the environment, for example, in the seawater, in soil, in sand, and also in slough. So this method involved this pyrolysis gas chromatography and coupled to mass spectrometry, or the thermal desorption proton transfer reaction coupled to mass spectrometry. So there are also other different kind of analytical methods. So here we see the first report about the NPs in the environment in the 2017. So we see here in the seawater, there are different kind of nanoplastics from PE, polyethylene, PP, polypropylene, polystyrene, and polyvinyl chloride, and also PET. So that means nanoplastics are really present in the environment. So therefore, there are some concerns about this bioaccumulation of MNPs in an organism, and also the transfer along the food chain. So they think this plastics self and also the additives in the plastic debris can be accumulated in the zooplankton, in bivalves, in the ocean, for example, and then it can be eaten by the fish, and then fish can finally come to the table of the human beings. So therefore, we think about this emerging threat of the MNPs to the food security and also to the human health. But except for the additives in the plastics, there are also other coexisting contaminants with these MNPs. So because MNPs has a very higher specific area, especially for the nanoplastics, and they are also normally strong hydrophobicity. This hydrophobicity can absorb many organic pollutants from the environment. And if these plastics are aged, then a lot of hydrophilic groups could be produced on the surface. And in this case, this hydrophilic surface can absorb heavy metals. For example, here is toxic bly, toxic copper, and also toxic cadmium. So that means the nanoplastic and the microplastic in the environment could be a source for the contaminants. This source comes from the additives, and then it could be also a sink for the contaminants. That means you can absorb and accumulate pollutants from the environment. So this accumulation of the pollutants on the MNPs is the absorption, via the absorption, and the mechanism for the absorption could be hydrophobic action, electrostatic action, van der Waals force, hydrogen bonds, and the pi-pi interaction. So these mechanisms, I think you have already known from the other talk about the absorption of pollutants on other nanomaterials. So they all have the same mechanisms. So this absorption of pollutants on the MNPs has great impact on the bioavailability of the pollutants, on their persistence, on their mobility, and also on their bioaccumulation in organisms. Yeah, because this absorption can increase this concentration of pollutants in the water, in the environment. But these adsorbed pollutants on the MNPs can be desorbed or can be released into the environment again if the environmental conditions change. So if these pollutants from one place to the other place transported by the MNPs, then in the new place, these pollutants are released into the environment again, and this works like this train house. So this is a kind of train house effect. So the factors influence the adsorption of pollutants on the MNPs. That means it can influence the train house effect. The factors are the following. First one is the MNPs properties self. So this polymer types, this MNPs size, the functional groups on the MNPs, and also the wearing intensity of the MNPs, the surface area of the MNPs. This is closely to the particle size, and also which kind of additives in the plastic. And the last one also very important is the crystallinity of these MNPs. So this one is different from other nanomaterials. So in MNPs, here they have crystallinity, and other nanomaterials maybe they don't have this property. And also the pollution properties have effect on the adsorption. So for example, here, what kind of functional groups of the pollutant have? How about hydrophobicity? How about their molecular weight? And also the concentration of the pollutants play a role for the adsorption. And the last, of course, is very important, is that environment conditions can have a great influence on the adsorption and the desorption of the pollutants. So for example, the ionic strength, the pH value, the dissolved organic matter in the environment, the surfactants present in the environment, and the redox state, and also the temperature plays a role. So all this can influence this adsorption, of course, also the desorption of pollutants on the MNPs, and therefore also this transport of these pollutants in the environment, and also the joint house effect. So this is a basic introduction about the MNP or the nanoparticles in the environment. And then I would like to show you two studies in my lab to show this the effect of nanoplastics as vector. So the first one is mobility and vector ability of nanoplastics in the saturated soil. That means the soil is saturated with water. That's a porous medium. So we did such experiments. So we put this nano plastics, is polystyrene nanoplastics, PSNPs, and also with pollutants. These pollutants are here. Below is pyrene, bisphenol A, 4-nonylphenol, bisphenol F, and also the BDE 47. So there are different kind of pollutants with different polarity. For example, the pyrene is non-polar, and nonylphenol is polar. So we put this together on the one soil column. So this experiment for the vector experiment was performed with the soil column. So we put this together on the soil, and then we pump this electrolyte through the soil, and then we collected the effluent and analyze the concentration of different pollutants, and of course also the concentration of the NPs. So the pollutants was radioactively labeled, so it's easily to detect the quantification, and the NPs, the nanoplastics, is fluorescence labeled, so they are all labeled. So and also of course this NPs without the presence of pollutants are also done with on the column. So here is the results. There is a first one here. We see this transport of this nanoplastics through this column. This curve is a broke-through curve. So we see here the PV is pore volume is effluent volume. So you see here this NPs was eluented, and are collected, detected. And this peak is also not a real peak. It's because as experiment we cannot avoid this peak. So just forget this peak, and here is this breakthrough curve. So we see here at the different concentration of the PSNPs. We see here below is a different concentration, is 0, 5, 10, and 20 milligram per liter PSNP. They can all elute from the column and at the same rate. But this organic pollutants, the Pyrene, eluted with different rate. So here, if there's no PSNPs, this red one, so there's no elution of the Pyrene. If this PSNPs concentration increase, and then this Pyrene was eluted from the soil column, and also the concentration increase. That means here we see that the Pyrene was co-transported with this nanoplastics. And the higher concentration of the nanoplastics, the higher amount of Pyrene was eluted. So this for the Pyrene is in this case, and this is for the BDE47 is also in this case. That means these two organic pollutants can be eluted, co-eluted by the PSNPs. And in contrast, we see here the other three organic pollutants, BPA, this BPF, and this nonylphenol, are not affected by the PSNPs. That means in the presence and absence of the PSNPs, they are all eluted from the column with the same amount. So that means here is the conclusion. Conclusion here is PSNPs increase transport of Pyrene and BDE47, and they have no effect on the transport of BPA, BPF, and nonylphenol. So why could it happen? Then we studied this could be owing to the desorption of pollutants from these PSNPs. We see here is the desorption. Here is sorption, and this one is the desorption here. So we see Pyrene and the BD47 could not be desorbed, or this desorption is not completely reversible. They had one hysteresis index. So the higher this HI, that means that the desorption is more difficult, so less desorption. So the Pyrene and the BD is less desorbed, and therefore we can understand that they absorbed stronger on the PSNPs. That means therefore they can be co-eluted with the NPs through the column. But here the other three organic compounds, BPA, BPF, NPs, we see here they are all on the same lines. That means this desorption is almost completely reversible. So that means the BPA, BPF, and the NPs on the nanoplastics can be completely desorbed in the soil volume column. Therefore, they could not be co-transported by the NPs through the column. They remained almost completed on the soil particles. So here should be, yeah. So there's a desorption hysteresis determines the vector effect of PSNPs. So why? So we see here, here is some polarity and also locale W with here the Pyrene and BDE47. They are non-polaric and they have stronger adsorption on the pollutants. Yeah, therefore, you can understand they are very strong and the absorption, therefore the desorption is less. But here for the NP, nonylphenol, the absorption of nonylphenol on the NPs was also very strong, the 70%. But the NPs was completely the adsorption of MPs was completely reversible. Why is MPs is reversible? So then we know that it depends on the structure of the polystyrene plastics. So polystyrene plastics with this chemical structure, and it is difficult, this chain could be packed in the crystalline form. And this means is the polystyrene plastic is in amorphous state. And amorphous, there are a lot of pore in the inside of the plastics. And therefore, these non-polar contaminants like pyrene and BD47, they can diffuse into the pore of the PS. And in this case, they have stronger adsorption, and this diffusion from inside to the outside is also very slow, and therefore the desorption was very slow, is irreversible. Therefore, they behaved as this adsorption hysteresis. And for the other compounds, the BPA, BPF, and the nonylphenol, they are hydrophobic with a hydrophilic hydroxy group here, and they could not diffuse into the pore, and therefore they are adsorbed only on the surface. So on the surface, therefore, they can be dissolved in the soil environment, and therefore the nonylphenol, even nonylphenol, had a very strong adsorption on the particle, but the desorption of the nonylphenol was also very strong, and therefore the co-transport of nonylphenol is very low, like this case. So this is the co-transport or the vector effect of nanoplastics in the pristine form. I had said at the beginning that plastics environments can undergo the aging process. This aging process can change the surface properties of the plastics, and therefore we want to know how about this aging process influence this physical chemical, can influence the mobility of the MPs self, and also how they influence the ability of the MPs to transport the pollutants in the soil. So for this, we did this experiment. Firstly, we prepared the aged nanoplastics under UV irradiation for 6 hours and 20 hours respectively. Then we got this UV PS1 and UV PS2 plastics. And also the salt plastics is treated with ozone, and we got ozone plastics. So that we got 3 plastics, is 3 different of nanoplastics with aging, aged, and this is the original without aging. We see here, this aged plastics has a smaller size, they are smaller, and especially for the UV PS2 plastics, they are aged for 12 hours. Yeah. So this size is become smaller, and here the surface also rougher. So here is also rough, but it's not so clear here, but they are all rougher. For the UVSP2, this change was more significant. So with this 4 plastics, nanoplastics, we did the same column experiment as before. So we see here, this is a illusion of the different plastics. The red one is the original PS, and this is the UV PS1, UV PS2, and this is the ozone PS. So we see here these different plastics could be eluted from the soil in the different electrolytes and different concentrations. And this ozone treated PS, this one, eluted mostly significant. So here is the aging significant increased mobility. Yeah, you see as aging, it's not aged, it's aged, increase the mobility of the nanoplastic. And this ability this order. I said this ozone 3, ozone treated PS has the highest mobility, and why the ozone treated PS has the highest mobility, we did a calculation. So it's according to the XD-LVU theory, and we found that the surface hydrophilicity of the MP is on the ozone treated PS is a high, increases the primary, increases the mobility, and this is the primary reason for the mobility increase. So this is a surface hydrophility plays a primary role for this mobility. And then we checked this vector ability of edged PSNPs. So we see here, this is a plastic self, and then this is a pyrene co-eluted with the PS. We see here with the, here is a control, and this one is original PS, and this is treated aged PS, and we see here the aging also significantly increases the vector ability of the MPs. We see here for pyrenes, this case, for the nonylphenol, also this case. Yeah, we just did two organic pollutants. And this mobility of MPs is the same. This is the last slide that showed the ozone treated PS has the highest mobility, but the co-elute of the pollutants was in other order. So the the highest is the UV PS2, is not ozone. So that means they are not in the consistent. That means the highest highest mobility of nanoplastics could not have the highest vector effect, vector ability for the pollutants. As here, there's a different. And why could it cause this case? Then we thought this could be related to the adsorption affinity of the pollutants on the edged nanoplastics. We see here this pyrene on the different plastics. We see here the aging has no significant effects on the pyrene adsorption. So here they are closely together, but aging has significant effects on the absorption of nonylphenol. So that's with the aging, that's nonylphenol adsorption on the particles increased significantly. See here, it's original, and here is significant. And this UV PS2 has the highest adsorption affinity to nonylphenol. So that could explain why nonylphenol can be co-eluted by the UV PS2. So that here, firstly, we know for the last experiment that nonylphenol can strongly adsorb on the PSNPs, that is hydrophobic interaction. But after this aging, after the aging on the surface of the MPs, there are also many oxygen content groups, for example, the hydroxy groups, and they could have this hydrogen, and this hydrogen can form the hydrogen bonds with the nonylphenol. So with nonylphenol had also one here, hydroxy groups, and they can form these hydrogen bonds between nonylphenol and the nanoplastics. And in this case, the adsorption of nonylphenol on the UV PS2 was higher. And then we see here how about the desorption of the pollutants from the edged MPs. So if the desorption is very high, then this pollutant could not be co-eluted from the soil. So here is the results about the desorption of the pyrene. This is the pyrene, and this is a nonylphenol on the original PS, and then this on the PS1 and the UV PS2 and ozone treated PS. And we see here that's the here. Thus, we see here this moment. Yeah, the nonylphen‌ol aging increase the irreversibility of the adsorption. So here we see here this not on the same linear, it's also on the left side. It means it's a desorption hysteresis. We calculated here. Here is calculation. Here this aging increase this irreversibility of the adsorption. Both here's both the nonylphenol and the sorry. Yeah, it's only the nonylphenol. Here is only the nonylphenol here. On the nonylphenol here, and this increase the irreversibility is owing to the hydrogen bonding between the nonylphenol and the MPs. I said introduced the last slide. And here when we see here the yellow digital is for the pyrene irreversibility. So this is high. This higher is this means that the irreversibility was not highly effective. So here means we see here this after the calculation we see that aging had no significant effect on the irreversibility of pyrene adsorption on the MPs. So that means aging increases the adsorption of nonylphenol on the MPs and also increases the adsorption irreversibility. That means the nonylphenol can be co-eluted by the MPs from the soil. And this pyrene, the irreversibility of the pyrene was not effective, and therefore the pyrene could be also theoretically could be dissolved in the soil. Yeah, but this pyrene was also co-eluted by these MPs. Why? They could co-eluted by MPs and they are not dissolved. And here we see that because this polystyrene nanoplastics has a lower crystallinity, and they have the pore in the particle. And after this aging, the pore size increased. We see here is original, and this is after pore size increase. And this increased pore size can absorb more pyrene, and this pyrene in the pore is difficult to diffuse out, and therefore this desorption of this pyrene from the aged nanoplastics is became slower. Yeah. So this is the adsorption hysteresis we see here. So this is adsorption and this desorption. This desorption is very slow. Dynamically, we see it's very slow, and here this a kind of hysteresis. And in the case of nonylphenol, we see here this desorption of nonylphenol, and the desorption very quickly, decrease very quickly. So this here, in compared to the nonylphenol, this adsorption of pyrene has a stronger hysteresis, and this stronger hysteresis contributed to the increased vector effect of MPs to the pyrene. So this owing to two reasons. One is the micropore increase, and then is the structure alteration. The structural alteration we see here is surface area, molecular weight change. This is our structural alteration. This structural alteration is also a reason for the pore size change. So there is a summary. Ability of PSNPs to mobilize, transport micropollutants in saturated porous media. They are contributed to the micropore and to the surface adsorption. So this is for the non-polar is microfilling. Yeah, and for the polar is to the surface adsorption. Yeah, for the non-polar also the surface adsorption. So it's like here. And the aging of the microplastics can increase the mobility of the plastic itself. This is owing to the increase in the hydrophility, surface hydrophility of the nanoplastics. The aged nanoplastics had a higher ability to transport micropollutants owing to the several reasons. One is for the non-polar pollutants. The non-polar pollutants is adsorption hysteresis, and for the polar pollutants is a reversible adsorption via hydrogen bonding. So, Doc, I see the time is almost over. I skipped this part two and then direct to the take-home message. Yeah.
Yeah, that's good.
Okay. So MPs, we say the MPs are different. The nanoplastics are different from traditional nanomaterials. The traditional nanomaterials are manufactured specifically with specific goals. And these MPs are mostly secondary products in the environment, secondary products of the plastic debris in the environment, or produced during the use of plastic products. This is the first one. The second is the pore filling and hydrogen bonding played an important role in the vector ability of MPs. And the pore filling here is also different from the adsorption of organic pollutants on the traditional nanomaterials. The third one is the size of the nanoplastic may play a very important role. So the nanoplastics may have a higher biological toxicity. I just omitted this part, but I said this here. So the smaller nanoplastics may have a higher biological toxicity and stronger effects on the environmental behavior of the coexisting pollutants. So they could increase the bioaccumulation and the persistence. But the unknown question is, how about the lowest limit of the size? Because we know when the size decreases, then these plastics, MPs, will not be particles. They could be dissolved organic matter, like a humic substance, for example. But how, or where is the lowest limit of this size for the nanoplastics, which has the toxic effect? We don't know now. And the fourth one is about the health risks of the nanoplastics and their carried pollutants. This health risk to the human beings and also to the other animals are still unknown. And these unknown questions need to further investigation. So then I come to the last slide of my presentation. It's the acknowledgement. This experiment was done by Dr. Yili Ma, now professor in Hainan University, Dr. Liu Jing, now in another university in Tianjin, and also the Professor Chen Wei, we co-supervise together here. And the funding agency is the Ministry of Science and Technology of China, and also the National Science Foundation of China are also acknowledged. And thank you for your attention.
And thank you very much, Ron, for a very interesting talk. So I would invite the audience to go ahead and ask questions. And I see that we already have one from Ian Oliver. I'm just going to bring it up so I can read it. Or Ian, maybe you just want to go ahead and ask your question.
Okay, thanks, Doug. Thank you, Professor, for your talk. I was thinking about, you showed that these nanoplastics, microplastics, the ionic strength is very important. So do you think that that means that because ionic strength influences sorption on the nanoparticles, you think that transport of these organic pollutants like pyrene is going to be a bigger problem in freshwater compared to seawater?
Yeah, it's a
Will
very
they
good
absorb
question.
more in a freshwater situation than the sea?
Yeah, that's very good questions. So in the freshwater, the ionic strength is low, and in the when the nanoplastics carrying pollutants from the river to the seawater, and then the change of the ionic strength could make big change on the adsorption. So this is one is maybe this pollutants could be desorbed from these particles, and also another possibility is that nanoparticles could aggregate together. So they could be homoaggregate or the homoaggregation. Yeah, they could also aggregate and then become bigger and then set down to the sediment on the estuary. Yeah, could happen. It's a very interesting question.
Thank you. Thank you. It's
Thank
good.
you.
Other questions for Ron? Maybe I'll ask one while people are thinking about that. It seems to me that the information you've presented today is sort of a good news, bad news sort of situation where obviously there are some situations where it would create some real problems, environmental problems, but do you think that you can customize nanoplastics for use, for example, in water treatment systems? And if so, would you be able to recover the nanoplastics, or is that not likely to ever work?
Yeah, it's also a very good question. So this is the some my colleagues are studying now in China. They want to do the removal of plastics, yeah, microplastic, and of course also nanoplastic from wastewater. So they did some experiment with wastewater, because in the wastewater treatment plant, yeah, microplastics are detected in the effluent. Therefore, they want to do the how to say higher, the further, how to say post treatment of the effluent. And yeah, some people use the, how to say this one chemicals to increase the size of these plastics. So this is a coagulation, process to increase this. And some I think this nanoplastics is not so easy, yeah, to get together. And yeah, maybe this put some coagulation to the wastewater. They could increase the size, and then they can sediment, and they can remove it. But the efficiency till now, I think it's not so good. My colleague has not tell me that they had a good results. Yeah.
Okay, thank you. Another question from Ian Oliver. What method do you recommend for identifying and quantifying these nanoparticles, plastic nanoparticles in organisms like shrimp or krill?
Yeah, so there are many identification methods. Firstly, these MNPs should be isolated from these organisms, from the tissue. And for this isolation, there are some methods. Most of them are digestion. So for the digestion, you can use, for example, acidic, like nitric acid. You can use peroxy HO2, H2O2, so like this strong chemicals. And we have another method. We use enzymatic digestion. So with this enzymatic digestion, it's a weak digestion process, and this modification of the plastics could be avoided. So with a strong digestion, then this surface of the microplastic could be changed, and then it could bring problem for the further identification. So this first is the isolation, and then the identification can use some spectroscopic instruments like FTIR, Raman, and also many other the optical based on the optical method, and also some colleagues using MS method. So they oxidize the pyrolysis these isolated particles, and then get the fragments, and using the fragments to identify which kind of MNPs in the tissue. So this is for the identification. For the quantification of the large size plastic is not a big problem. You can counting on the microscope. You can also using the MS method to quantify it, but for the nanoplastics, it's not so easy at the moment. There are also some methods, but it's not so easy from this organism tissue. It's not so easy. Normally, they need to concentrate it, firstly concentrate it, and then counting on the microscope. Yeah. Okay.
Maybe I have one more question for you, Ron. This isn't not so much a question, but sort of a where do we go from here question. You've shown some very clear evidence for the complexity of the situation for one particular type of nanoplastic. And now when we think about that 80% of all plastics that ends up in the environment, and some portion of that ultimately degrading to these small particles, what do you think the future holds in terms of being able to predict the fate of pollutant bound to nanoplastics? Do you think we'll be able to make some general predictions about certain classes of plastics compared to certain classes of pollutants, or do you think the situation is just so complex that it's going to be very difficult to to build some sort of a general predictive framework?
Yeah, it's a good question. So of course it's complicated, but I think if we have enough research on the different kind of plastics, and then we can get should get a general knowledge about this desorption of these different kind of chemicals on a specific type of the plastics. So this is like this, how to say, in some toxic areas, they use computers, they use modeling, and they can simulate the interaction between organic pollutants with macromolecules like protein and DNA, something like this. And I think this could be also used for the interaction between organic pollutant and microplastics. Yeah, but microplastics is more complicated because in the environment, the nanoplastics is not a nanoplastic itself. It could absorb many other macromolecules. This is so-called eco-corona, yeah, on the surface of the microplastic. And in this case, the situation will become more complicated. So, but I think this could be a research area. Yeah, good idea.
believe you're on mute, Doug.
I'm sorry. Thank you, Ian. So I see we're at the end of our time. I will give one last chance for any questions, but if there are no other questions, not hearing or seeing any, then Ron, let me take this opportunity once again to thank you for an excellent talk. You're obviously doing some really important work, and it's really interesting. I should also mention that Ron will be joining us as an invited speaker for our Science and Policy workshop that we're holding in October. So once again, I would encourage you to visit the website and to check that out. I want to also just mention that next week our speaker will be Professor Martina Wever from Leiden University in the Netherlands, and she's going to be speaking on nanoparticles in ecosystem effects of nanoparticles, I guess would be the best way to describe it. So I hope you will join us for that talk, and I hope you will also invite other people that you know who might be interested to join us as well. Also remind you that these talks are recorded and are available on our website, so you can go back and listen to them, or if you know anybody who has missed a talk and would like to hear it, they can go back and listen to the talks from the website. So once again, Ron, really appreciate you giving us talk today, and thanks to everyone else who's joined us. Hope to see you next week.
Thank you, Doc, for the invitation, and it's my great pleasure to give this talk. Thank you.
Thank you very much.
Yeah.
Bye to everyone for now.
See you later. Bye-bye.
Bye.